Thank you very much for the invitation to this very fascinating and very timely workshop, I think, as well. I'm interested in sex differences scientifically, but also very committed to gender equality, so I'm enjoying it in more ways than one. The lab hasn't interested in sex differences, but I guess overall, so this is going to be a hardcore science talk, so I'm going to try to keep it simple and hopefully a bit entertaining. So the lab has an interest. So we're interested in sex differences, but a kind of broader interest, I'd say, is to do with how organs maintain physiology. So we're multicellular organisms, we've developed organs, and these organs have specialized and they carry out different functions, right? But for all this physiology to work, all these organs need to work together. So we're interested in this kind of integration of organs in the context of physiology, and also how they change, how they change when they have to change, how they drive adaptive change, right? So those are the sort of general questions that we're interested in. So for these organs to work together, one thing that they need to do is to communicate, and one way in which they will communicate will be through exchanging signals. So these signals can be hormones. They can also communicate via neurons, so they can, there's neurons in all these other organs and they'll be sending messages. And now we also know that they can communicate through metabolites, so they can also exchange metabolites in the circulation, and that can have long-range effects. But the other thing that these organs can do is they can change themselves, so they can undergo pretty dramatic adaptations. Hopefully you've never come across a python before, but if you have, you might know that it has these very extreme feeding patterns whereby it'll go without food for about 2 months, and when it finally feeds, it feeds in style. So this is the python after it's ingested a small crocodile. What you cannot see from this X-ray is that the gut in this, so the intestine of this python, within 24 hours increases massively. And it's not just because it's got the crocodile inside it, everything is changing in that intestine. The stem cells are dividing, the cells are increasing in size as a result of this event. And then over time, these will go back to normal. So after after a sort of few days, over a month, the intestine will go back to its usual size. So you can argue this is a pretty extreme example, but we face sort of similar examples ourselves. So this is what happens, during pregnancy, during reproduction. We're all obsessed with that bump there, but what we're not aware of is that our internal organs are also changing pretty dramatically. So organs such as the liver, the pancreas, the intestine are changing. They're growing as the baby grows, and then to some extent they'll go back to normal. So that kind of plasticity is what we're sort of the kind of plasticity that we're interested in. So we've been exploring this plasticity in mainly sort of 4 different contexts. So today I'll tell you about sex differences and a little bit about reproduction. But we've also been exploring this organ plasticity in the context of nutritional challenges, and also in the context of changes in vascularization to these organs. And the other thing I should say is this is kind of somewhat unusual. We've been hearing a little bit about mice, but most of the work that we've done so far is in fruit flies. So you might be surprised to hear that we can use fruit flies to study this kind of thing. So fruit flies historically have been used for to kind of understand how animals are made. So this is about over 100 years of Drosophila research. So this is what Drosophila research looked like 100 years ago. There were only men at the time. Now we have women in the fly rooms as well. What's really useful about flies is that combination of things. They have this very short generation time. We can breed them very rapidly. We can very rapidly turn genes on and off. We can do it hundreds at a time and see what happens. And we can, and 65–70% of these genes will be conserved between flies and humans. So whatever we can learn in flies, and we can explore, move on to mice or move on to humans and see whether we find similarities. So that's been really powerful. So this kind of research in flies has led to 6 Nobel Prizes, most recently last year on circadian biology, circadian clocks. So that was the kind of research that had been done for many years, and that was really mainly to understand how animals were made. Flies had not really been used to study physiology until relatively recently. And what kind of changed the game was the identification of insulins in flies, perhaps now 15 years ago. So flies don't have pancreas, but they have insulins, and they have all sorts of hormones that regulate sugar levels, how fat they get, what they choose to eat. So now we know that flies can get diabetic, they can get obese, they can make nutritional choices that are pretty sophisticated and will change their food preference depending on a number of things, depending on internal state, depending on whether they're laying loads of eggs. So there's still pretty sophisticated control of all these physiological decisions. So we've been using the fly to explore physiology, and in particular we've been focused on the intestine of a fly. So flies have intestines, and the intestines don't look that different from ours in the sense that they have stomach, they have a small intestine, they have a large intestine. And if you zoom into this, if you zoom into this intestine, we'll find cell types that we would find in a intestine. So this epithelium, so the lining of this gut, will have stem cells, and they will give rise to enterocytes, which are the digestion or other cells that can secrete hormones into the circulation. But the real advantage for us is that we can genetically target all these different cell types in isolation without affecting anything else in the fly. And we can do it also temporally. So that's very useful when you're trying to explore tissue crosstalk and physiology. So it was doing these kind of experiments that we came across these sex differences between males and females in the intestine of the fly. So we were doing a very simple experiment, and basically we were just dissecting intestines, and we were looking at what was different between them. And we were doing it pretty systematically. So we were looking at males, we were looking at females, we were looking at flies that had been exposed to an adipogenic diet, to a sugar-rich diet, and we were looking at what was different between all these different intestines. And by far what was the most difference was just males and females, just looking at them without doing anything. So really up to 10% of gene expression was sexually dimorphic in the gut, and up to another 10% was sexually dimorphic at the level of splicing. So overall really about 15% of gene expression. So 15% of genes were different between males and females. So that got us quite interested in sex differences, and that's how we kind of started this work. When you start thinking about sex differences, and that's something that we've already, so previous speakers have alluded to a little bit, you can kind of explain them away in different ways. So you can say, well, you know, maybe a male intestine and a female intestine are different because they have been made different during development. And indeed, there are examples of this in both flies and in mice. So the brain can be wired differently during development, and that will make the brain function differently. And indeed, if you just look at a male and a female intestine in an adult fly, you can see that it looks different because it has been made different during development. So that's one way by which these differences can arise. The other one is through hormones, right? So we've heard a little bit about this already. We've all gone through puberty. We know how powerful hormones can be, and that's really the sort of prevalent view in the kind of mammalian field, right? So we think that mainly the main differences between all of us, in mice, all of them are down to hormones, but as previous speakers have already alluded to, there's increasing evidence that there might be some contributions of sort of intrinsic sexual identity of sex chromosomes within cells. And I think a very nice example illustrating this, and that's something that happens accidentally in nature, so these so-called gynandromorphs. I don't know whether you've ever come across them. So these are bilaterally asymmetric animals, so these are naturally occurring. We haven't done anything to them, and one half is male and the other half is female. And that happens in flies and in butterflies, but also in chickens, for example. And what's very striking about these gynandromorphs is that the two sides are dramatically different in size, right? So if it was all about hormones, you wouldn't necessarily expect the two sides to be different. So we wanted to know where these sex differences were coming from, and how they sort of arise. Is it developmental? Is it adult specific? Is it through hormones? Is it through intrinsic mechanisms? And our work has focused mainly on three different cell types that make this intestine, right? So the intestine is an organ that's made up of different cell types. So we've mainly focused on intestinal stem cells. So these are the cells that replenish our gut. So our gut gets damaged and it needs to be replenished. So the stem cells that are constantly dividing to give rise to new cells. The other cell type is the enterocytes. So these are the progeny of the stem cells. So these are the cells in our lining that do all the digestion and absorption. And the other one is the neurons that innervate this gut. So this gut is populated by nerve cells, almost as many as in a spinal cord in the case of humans. And we know quite little about what all these neurons are doing. So we've also explored whether these neurons might have some sort of sexual identity. And I'm just going to briefly summarize what we found and what we're finding, and then I think I'll just give you a few examples of what I mean by that, because I think that might be a little more helpful. So really what we're finding, I think it's sort of pretty, so it's complicated, right? So even within an organ and within a very simple epithelium, there are many different ways to specify sexual identity. So we have 3 different cell types, and in these 3 different cell types, we find 3 different ways to specify sexual phenotype or sexual identity, if you like. And each one does it slightly differently. So in some of them is only intrinsic, in some of them is only extrinsic, and in some of them is a combination of both. What's increasingly clear, at least in the intestine, and at least for the readouts that we're looking at, is that it's not developmental. I mean, in all cases, we can reverse it in the adult. So we can allow a fly to develop as female and then change it genetically, and the cells will respond, right? So we can masculinize and feminize stem cells or enterocytes or neurons, and they'll still respond to these changes in the adult. So it's quite plastic, it's quite flexible, which is quite interesting. The other thing that we're finding is that each of these 3 mechanisms in the different cell types controls different sexually dimorphic features. So in the stem cell, sexual identity will control proliferation. In the enterocytes, I'll briefly tell you, it controls metabolism. In the neurons, it controls the signals that make these neurons eat more or eat less. But collectively, what we're finding is that they all seem to kind of adjust properties that affect the organ as a whole. So they somehow adjust organ level properties which collectively seem to optimize reproduction in males or in females. So I'll show you a couple of examples of what I mean by this, but what I would also like to convey is that these doesn't seem to be their only role. So we're finding very strong effects on reproduction, so the intestine needs to be male or female to maximize male or female reproduction, but we also find effects that are different, so separable from these reproductive roles. So just to give you a couple of examples, the first work that we did, and this is now published, so you can read more about it if you like, is on the stem cells of the gut. So one thing that I had very clear when we were looking at just simply gene expression between male and female guts is that the female guts had this upregulation of any genes that were involved in proliferation, cell cycle, etc. So that's what made us look at the stem cells, which are the cells that divide in the intestine. And what we found is that these cells invariably divide more in females. So they do it normally, so even in unchallenged circumstances, a normal homeostatic replenishment of the intestinal epithelium, and they also do it in response to damage. They divide more. So hopefully you can see it here. I'm showing you as green blobs, so wherever you see more green blobs means more proliferation. So the females, there's more green activity in the females. And then we can play these tricks in Drosophila, which are harder to do in mice, in which we can selectively masculinize or feminize specific cell types, right? So now we can do these kind of experiments in which we take a male fly and then we allow it to develop as male, and we selectively feminize the stem cells specifically in the adult. And now we can get these male guts to proliferate like female guts. And conversely, we can just take a female gut, which we allow to do. So in a normal female fly, we allow it to develop as female, and then we masculinize the stem cells, and that reduces the proliferation to male-like levels. So these kind of experiments suggested to us that there was an intrinsic component to this sexual identity, so somehow the gut stem cells knew their sex. And we've tested this in various ways. This is another way to explore it. So here what we're doing is something similar. So what we're looking at here is that a single stem cell and all its progeny within a window of time. And whenever we do that in normal males and normal females, and you can see a lot more green in the females, right? So this stem cell has been more active, it has made more progeny, and now we have a so-called clone, which is bigger in the female than in the male. If we now take a female and we masculinize these stem cells, we see that the clones shrink, so they're more like male clones, whereas of course we have no effect in males because the males were males to start with, right? So collectively these and other experiments told us 2 things. First of all, that the identity of these, so these stem cells knew the sex, so it's to some degrees or to a large degree cell intrinsic. And the other thing is that it's adult reversible, right? Because we're doing all these manipulations specifically in the adult, and we can masculinize and feminize the stem cells. I won't go into the molecular details of what we worked out there, but what we also found is that the mechanism by which this happened So the sex chromosomes are sent, so it's sex chromosomes, but the sort of how that is relayed molecularly turned out to be different from what we knew in flies. So it wasn't mediated through the normal or the canonical effectors of sex differences. So we found that there's a new branch of the sex differentiation pathway that sort of imparts these differences. And what's sort of rather interesting, perhaps more generically, is that the mediators of these, or at least some of the mediators of these sex differences, belong to families that are also represented in mammals. So we have these genes or similar genes to the ones that we found to mediate these differences. So conceivably they could mediate sex differences in other systems, not just in fly stem cells. So the question that we had, or one question that we had at the time, is why does a female gut need stem cells to proliferate more, right? So why does why do female guts need to be more active? Why do they need to divide more often? So one way in which we tested this is, so we did the kind of experiments that I was telling you when we masculinized the stem cells, specifically in the adult, right? So we have a female fly, we allow it to develop as a female, and then we masculinize the stem cells, and then we simply waited. We waited 3 weeks and see what happens. So what happens is this, so that's a female gut at the top, that's a normal one, the normal male gut. And now if we masculinize the stem cells of the female gut, over time the female gut becomes smaller, becomes more like a male gut. Whereas when we do it with the male was male, so nothing happened. In a way that kind of, so what that tells you is that the sexual identity is required to maintain a difference that was established during normal development, during the making of the animal, but it doesn't really answer why you want the female gut to divide more and whether this is actually meaningful in any context, right? But an answer that we found to this question, so we think that the more physiological reason why the female gut needs to be more active is to do with reproduction. So if you remember at the beginning, I told you that female guts grow a lot during reproduction, and the fly is no exception. So only 3 days after female fly has sex, the gut has increased massively. So 60% increase in the number of cells. So that's what's shown here. So this is the virgin gut, the mated gut. It's bigger not just because it's full of food, it's bigger because it's got many more cells. If you masculinize, if you now take a female fly in which you've masculinized the stem cells, this resizing doesn't happen, or it doesn't happen so well. So somehow the female gut needs this kind of built-in plasticities to allow the organ to grow during reproduction. And if we prevent this organ remodeling, we can do that as well. If we prevent this from happening, then reproductive output goes down. So it kind of shows that this resizing, this remodeling of organs is important to maximize reproductive output. So now I've presented it several times, and actually, I mean, that's something that maybe we should discuss tomorrow as well. I think one of the, maybe one of the challenges or something that's perhaps preventing us from studying sex differences is what we do with this knowledge, right? And I think that's something that I've experienced myself, so I've presented this data a couple of times, and I've had a couple of questions or sort of people in the audience kind of upset that I was kind of suggesting that the female gut was better than the male gut in some ways, right? So, you know, with statements like the female gut is more adaptable. I mean, I guess what I mean by that is that it's, you know, it can grow when it's needed, right? Whereas if you masculinize the identity of the stem cells, it cannot do it. The question, so I guess two related questions to this kind of statement is, well, is it always more adaptable? I mean, is it more adaptable in every possible way, right? Is it always that now if we expose that gut to a particular challenge, you know, it's gonna always be better at adapting? I think the answer to that is probably not. And it's emphatically no to the question of whether it's better than the male gut. And I'm just gonna show you an example of what I mean by this. And to me, all these sort of sex differences always have to do with trade-offs, right? And that's something that we should think about a lot, I think, when we convey messages about sex differences. And this is the trade-off for female flies, right? For female guts. So the female gut will proliferate more when it's needed, but it will also proliferate more when it's not needed. And that makes it more vulnerable to murogenic insults. So flies have been using, the fly gut has been used now for some time to model tumors, so we can induce tumors in flies, and typically people have used females. And they either didn't know why they use females, or they knew that kind of it doesn't work so well in males, right? And that's just a very clear demonstration. So now we have a control female fly on the left. Now we induce a tumor, and you can see this massive dysplasia after a couple of weeks in the female gut. We do the same experiment in the male gut, and you hardly see anything. And now we take a female gut in which we've masculinized the stem cells, and we can prevent tumor formation, like in a male one. So this is the particular trade-off for the Drosophila intestine. I'm not saying this is necessarily translatable to mammalian guts or to human guts, but I think it illustrates nicely this kind of concept of trade-offs when it comes to sex differences. And since then, in the context of stem cells, there's been work by us, but also by other groups that is showing how sort of different manifestations of this stem cell of this differential stem cell proliferation, right? So there's consequences, not just at the level of organ plasticity and tumor formation, which is what I've shown you, but also at the level of immune responses or how the gut reacts to age-related dysplasia. So collectively, male guts will be better for some things or worse for some things than the other way around, but that's something that we need to probably think about. So I just want to end also by showing you an example of what I mean by it's complicated and how different cell types do it differently, right? So all the stuff that I've told you was about stem cells, and there we have a very strong intrinsic component that controls how much these cells divide. We've also been working with with the enterocytes. So these are the progeny of the stem cells, right? So these are the direct progeny of the stem cells. And then what we find again is these very strong sex differences, and these strong sex differences have to do with how these enterocytes handle sugars. So sugar metabolism is very different in male and female enterocytes. What's really interesting about this is that I'm telling you enterocytes, but it's enterocytes in only one portion of the gut. So we have a whole intestine, but the sexual dimorphism is only in this portion here. So we have a digestive enzyme, for example, here in green. So this is the whole small intestine of the fly, digestive enzyme in green. It's not sexually dimorphic in the anterior region. It's massively sexually dimorphic in the posterior region. And we've done some work that now tells us that each region is key to sugar handling and seems to impact all sorts of things, including testes physiology. And mechanistically, again, what we're finding is that it's rather different from the stem cells. So it's not really about these enterocytes having an intrinsic sexual identity. So it's not the sex chromosomes deciding how they handle sugars, but in this case, it's a signal from the testis that's right next to it. So somehow the testis makes this particular bit of the gut male, at least for the sugar handling. And there seems to be some crosstalk between the two. So that's in flies. So you might argue, well, this is all very well, but like this is all flies. I'm sure my gut is not sexually dimorphic. That is not true. So I think there's evidence now from both mice and humans that it is. We are doing some of this work ourselves in mice. So we started in the same way. So we typically use a fly to discover and then the mouse to explore whether there might be some evolutionary conservation. And we've been doing this now for a couple of years and for things that are not necessarily sex-related. But in the context of sex differences, we simply started by looking again at gene expression, right? So what was different between a male small intestine, a female small intestine, and in this case, a lactating small intestine, right? So a mouse that's been pregnant and now is lactating, what's different? And again, you can separate all these three types of gut. So you can very clearly get a male signature, a female signature, and a lactating signature. And then the question that we have and that we're trying to address is how many of these differences are hormonal and how many of them are intrinsic. So one way in which we're going about it is, and then the other thing I should say as well is, so this is work that had been done, so we're revisiting these reproductive changes. So we are not the first to see these lactating changes. Back in the 60s, people had already described this pretty dramatic transformation of the small intestine of mice. Sort of at least at the anatomical level. So we're revisiting this with these modern techniques. So then again, we're trying to understand how many of these are intrinsic, how many are extrinsic. The way we're going about it is partly through organoids, right? So we're deriving small intestinal organoids from the mouse. We have them outside in a sort of more sex hormone-free, if you like, environment, and then we can genetically manipulate the sex, so the sort of sex chromosome genes, right? So we can begin to parse what's sort of intrinsic versus hormonal. They retain some sort of sexually dimorphic signature, so we can tell male and female organoids apart, which for me is quite exciting. So now we need to see how many of these differences are because of sort of epigenetic memory of hormonal exposure and how many of them are because of sort of intrinsic contributions of sexual identity. Sorry, that's. So I think I'll just end by trying to sort of summarize what we know and where sort of what we would like to explore in future. So I've told you that the adult intestine is sexually dimorphic, so it looks and functions differently in males and females. This sort of gut sex helps the female gut grow during reproduction, so when it's needed, but at the same time it makes the males and the females differentially susceptible to tumorigenic insults. For me, what's quite interesting or perhaps quite surprising is that different cell types within an organ know their sex in different ways, so that's through the sex chromosomes, through the hormones they listen to, and that manifests itself in different ways too. I think what we perhaps need to explore looking forward is what the relative contribution of all these different intrinsic versus extrinsic mechanisms are, and in particular also how plastic they are, right? Because so we tend to think of sexual identity as something quite static, right? So you're male or female, but all these things could be modulated, right? They could be modulated by diet, they could be modulated by internal state to some degree. So I think it's also quite interesting to explore the plasticity. So we know that we can turn males into females and females into males, specifically in adults. The question is, do they normally do that in any circumstances, and do they become more male or more female, and for what kind of purposes? So we're quite curious to explore this. And I guess a related question is why are there so many ways to specify sexual identity, right? So there's sex chromosomes, there's hormones, and I guess you can view these in two different ways. So you can say, well, you know, surely you want to ensure robustness of sexual identity to make males really male and females really female to ensure that they reproduce. But at the same time, it could be a mechanism to allow for some sort of plasticity, right? So you may want to tweak intrinsic mechanisms or extrinsic mechanisms in certain circumstances to provide some sort of plasticity. So yeah, so we're quite curious to explore this. Last but not least, to acknowledge all our funders, and especially the people that did the work. I didn't do any of this. So all this work was done by former postdoc in the lab who just left to set up his own lab in France, and he's going to be looking at sex differences as well. So Aneva de Goya helped a very talented master student as well. And I think that's all. Thank you for your attention.